Good morning. First, I'd like to thank the meeting organizers for the kind invitation. It's wonderful to be here. What I'm going to do today is to tell you about our recent attempts to develop more effective treatment for persistent Lyme disease. So I'm going to start with an overview of the complexity of the Lyme disease and the bacteria that cause the disease, and then tell you about the persistence problem of Lyme, and discuss the different possible causes for PTLDS condition, and then tell you about the work on Borrelia persisters, its mechanisms, and the limitation of the current Lyme antibiotics against such forms. Then I will use the TB treatment as a model for developing more effective treatment for persistent Lyme disease. As you will see, there's a lot of relevant similarity in developing treatment for persistent Lyme. So then I will tell you about our work on identifying drugs targeting Borrelia persisters, and then the use of the drug combination approach to more effectively target Borrelia persisters. And then tell you about our work on some essential oils that show amazing activity against Borrelia persisters, and then tell you different strategies for treating persistent Lyme, and ending with future directions. So in this audience, we all know that Lyme disease is the most common vector-borne disease in the US and in Europe as well. In fact, I just came back from a trip to France where I was invited to talk about Lyme treatment. There I learned, in fact, in France, it's a significant problem as well, where 55,000 cases a year, that's equivalent to on a population basis, equivalent to the US. So it's a significant problem in Europe, in France as well. So there's a similar sort of a problem we're facing in the US, where we see the two different camps. And, there are physicians who say that it's really easy to cure, easy to diagnose, and the other as well, all familiar, the persistent problem with the Lyme. So the disease, as you all know, is actually rather complex. It's a very heterogeneous disease. During the early stage of the disease, it's pretty simple. It's a localized EM rash, then developing into early disseminated form, then late disseminated arthritis, then PTLDS. So it's a continuing spectrum where the IDSA seem to focus mainly on the acute active phase of the disease, which is relatively easy to treat. That can be taken care of easily by 2 to 4 week antibiotics. Whereas when the disease gets into the persistent phase, they don't respond very well to current Lyme antibiotic treatment with a single drug, doxycycline or amoxicillin or cefuroxime. As we all know, the problem with the persistent Lyme is that they don't respond very well to current Lyme antibiotic treatment, where it's about 10 to 20% of the Lyme cases don't respond well. Despite proper treatment with antibiotics, they continue to suffer from fatigue, pain, joint muscle aches, brain fog, etc. They're called the PTLDS, we all know. So there have been different clinical trials, as we all know, where there are two types of trials, where one type of trial showed absolute no effect. The Klempner study, which used IV ceftriaxone for 4 weeks, followed by 2 months of doxycycline, compared with placebo control, see no effect in improvement in symptoms. Then this Brian Fallon study, as well as Lauren Krupp study, showed that IV ceftriaxone had some effect in improvement in symptoms, but it's not a cure. It's only a temporary improvement. Then the symptoms can come back. Then, of course, there's a more recent Dutch study where it showed this has a very unusual study design. All patients treated with 2-week IV ceftriaxone, then divided into placebo or treated with doxycycline or this drug combination. where they saw no effect. Of course, all patients are already treated with 2-week IV ceftriaxone, so they don't really have a proper control group. So it's making the interpretation difficult. So in this Dutch trial, where then they see no improvement, I think there have been different reasons that can be responsible for this observation. One is that the type of drugs used. We now know that the current Lyme antibiotics don't work very well against Borrelia persisters. The second point is that whether the treatment length is sufficient enough for a persistent disease like a PTLDS condition. The other is the heterogeneity of the disease. When designing such clinical trials, it is critical that we design the trials properly. I have to really, because it's such a heterogeneous disease, then all these factors can affect the outcome of the trial results. So in future clinical trials, I know there being a lot of interest, like Dr. Horowitz, along with some other physicians, are very interested in clinical studies trying the persistent drugs. I think it is great, and it's important that we design the trials properly, taking these 3 points into consideration. One is persistent drugs, the other is how long we treat, the other is patient heterogeneity. As you all know, that the current treatment don't work very well. There is no FDA-approved treatment for PTLDS condition. Patients try all sorts of treatment modalities. It's all over the place. They don't work very well. It's like a Wild West. You see all kinds of treatments from antibiotics, herbs, IV, IG, hyperoxygen, etc. So what are the causes for the PTLDS condition? There have been different theories. One is the host response to dead antigenic debris. The other is autoimmune. The third is that host has produced residual damage during the infection process, so that it can be helped. The fourth theory is that due to co-infections with Babesia or Bartonella, etc. Then the last is persistent infection. So in fact, if you look at all this, there's a sign of persistent infection that's actually going through all these possibilities. That is, this host response to antigenic debris could well be due to viable but non-culturable forms, which Dr. Ambers talked about, as well as autoimmune condition. The Alan Steer's group published an interesting study where Lyme arthritis, some patients with Lyme arthritis go on to develop rheumatoid arthritis. So this Lyme infection can really turn into rheumatoid arthritis, autoimmune disease condition. So this shows this chronic inflammation, chronic infection can lead to autoimmune disease condition. So all these possibilities can potentially overlap. They don't actually exclude each other, can overlap in patients. So the organism that causes the disease is rather unusual. The Borrelia bacteria develop different morphological variants, starting from the spirochete, which we are more familiar with, but that may not be the most important form involved in disease pathogenesis. In fact, when it develops in the older culture, it changed from spirochete into round body forms, as well as aggregated microcolony form. When the culture gets older, gets into larger biofilm-like structures. These actually have implications in terms of response to antibiotic treatment as well as disease pathogenesis. So these forms are much more difficult to treat, much more tolerant to antibiotics, and more tolerant to host clearance by the immune system. So we actually can divide, we propose this new model called inoculum-dependent severity of disease. This is really to show that the PTLDS condition can be heterogeneous, can be complex, can be broadly divided into two types. One is type 1, that is during the actual tick bite infection process, they can already introduce biofilm-like structures causing a persistent disease, so that despite standard antibiotic treatment, you don't cure the disease. Okay? That actually seems to correlate well with some clinical observations. That is, even though the patients are diagnosed early, treated early, you don't cure them. Okay? It's most likely due to this. During the very infection process of the tick bite, the tick can carry very heterogeneous Borrelia bacterial population. When it's inoculated with biofilm-like structures, they can cause a persistent form of the disease that don't respond to current Lyme antibiotic treatment. So that's called the early development type. It's called the type 1 PDLDS. The other type is what we are more familiar with. That is, patients are infected, not diagnosed in time, not treated in time, then from early infection, develop into persistent late stage of the disease, then it's difficult to treat as well. So this is just like a cartoon to show you this early Lyme. Sometimes it's actually easy when it's actually introduced with maybe spirochete form primarily, that's easy to cure. It's just like a small carrots, right? But sometimes it's unlucky. You're inoculated with the large carrots, like a biofilm structure, and you can't really get it out. Then there have been different studies showing the persistence problem. As we all know, that's being shown in mice, in dogs, in monkeys. And then the organism get into this VBNC, which Dr. Ambers talked about. It's a really intriguing phenomenon, very interesting. And also in human studies as well, there have been anecdotal reports of culture of Borrelia organism despite treatment. But the intriguing thing is that it's not a consistent observation. It's unlike TB, persistent infection. When the patient relapses, you can see the organism, you can culture the organism. But oftentimes with Borrelia, with Lyme disease, even though the patient relapses, you cannot culture the organism. That's why making the disease very difficult to diagnose, and very easy to deny. Okay? So we need better diagnostic tools and also better treatments for such condition. So we and Kim Lewis group, Monica Ambers group, we all showed that Borrelia in vitro form persisters tolerant to current Lyme antibiotics. So current Lyme antibiotics are good at killing or inhibiting the growing form of the Borrelia bacteria, but have a very limited effect on the stationary phase bacteria. So this is just some background on bacterial persisters. It's not new. It's being observed when penicillin was first introduced. It was found that penicillin killed 99% of the bacteria. There's also always small percentage of bacteria that's not killed. This small percentage of bacteria not killed, it's not due to genetic resistance, but rather due to phenotypic resistance, because when you remove the antibiotic, bacteria remain susceptible to the same antibiotic. That's why it's called a phenotypic resistance. So we use this yin-yang model to express this. So it's really easy to understand. There are two types of resistances. One is resistance in growing bacteria. That is what we're familiar with. Genetic resistance has a genetic basis. The other is resistance in non-growing bacteria. In this dark part, in this non-growing persister form, it's called persistence. Okay. So here, in fact, people know persistent bacteria, persister bacteria, involved in persistent infections like TB, urinary tract infection, and I think in Lyme as well, as we will show you. So here I will use this TB treatment as a model for developing more effective Lyme treatment. So yeah, in fact, Borrelia developed persisters, there's a molecular basis. So we have done RNA sequencing of Borrelia persisters and showing that in fact some pathways are upregulated. Very interestingly, the protease, ClIP-P protease are upregulated along with DNA repair, etc. And these pathways in fact share similarity to persister pathways in other bacteria. In addition to these upregulated pathways, there's also chemotaxis transporters all upregulated. There have been outer membrane proteins downregulated, and also ribosome proteins downregulated. So here, this TB treatment regimen, which is a drug combination with 4 drugs, azanazid, rifampin, pyrazinamide plus ethambutol for 2 months, followed by a continuation treatment with INH, azanazid plus rifampin for another 4 months. So in the TB treatment, the model is that you have 3 types of drugs. One is type like azanazid that kills the growing form of the bacteria. Then you have rifampin that kills both growing and non-growing bacteria. Then you have a pyrazinamide, PZA, that kills exclusively the non-growing persister form of the bacteria. You need all 3 types of drugs in combination in order to more effectively cure persistent infection like TB. And in Lyme, we need exactly the same sort of approach. So in TB, in fact, people actually have learned all the lessons. That is, they know that the importance of drug combination in the old days because they have 60 years of experience treating TB. So they know the importance of drug combination because in the old days, people know that if you just treat with single INH azanazid that inhibits the growing form of the TB bacteria, you get only 30% culture conversion if you treat for 3 months. And then if you use combination of streptomycin plus INH, you get a higher percentage of culture conversion to negative in the sputum. Then it also showed the importance of the length of the treatment. That's also very important in developing a treatment regimen. You have to know how long you treat. In the old days, treating TB with 3 drug combination, INH, PES plus streptomycin, takes 2 years to cure TB. Because if you treat a shorter time, for a year, you get 19% relapse. If you treat for 6 months, you get 62% relapse, very high relapse. That's why you have to treat sufficiently long. Okay? This was in the old days, in the 50s. Okay? But then, subsequently, TB people learned the lesson, then they know the idea, the concept of persister drug. That is, when they found they add rifampin, which is a persister drug, adding rifampin to this treatment regimen shortened the therapy from 2 years to 9 months. And more importantly, when they add pyrazinamide, which is a true persister drug, to this regimen, you shorten the therapy from 9 to 12 months down to 6 months. That is what we have. Without pyrazinamide, the therapy is longer. So that's why, that's all shows the importance of persister drug. So now in the TB field, we know, and then it's the same sort of a concept we need to extend to treatment of persistent Lyme disease. So this is the cartoon probably many of you have seen already. The current Lyme antibiotic is like this mower, which is only kills the growing, chop off the growing form of the bacteria. You still have the root there. And when you remove the antibiotic, stop the treatment, the whole thing grows back. What you need is a shovel, like a persister drug, that dig out the root, then you cure the infection. So that's why we want to really apply the PZA principle, or Yin-Yang model, okay, to the treatment of persistent Lyme disease, where you need all 3 types of drugs, where you need drugs like doxycycline that can be active against the growing form. And ceftriaxone is a drug that's interesting, that has activity against both growing and a non-growing form of Borrelia. Then we need persister drugs like we identify, like daptomycin, clofazimine, sulfa drugs, and all these. All these drugs have to be used in combination in order to more effectively cure the disease. So when we started, there were no drugs active against Borrelia persisters, so we have to develop a screen, cybergreen PI screen. We screen the FDA drug library, NCI compound library. These studies have been published. I'm going to be very brief, very quick with this. In the first study, we identified the daptomycin, clofazimine, cefoperazone, sulfa drug, etc., active against Borrelia persisters. Then published in the second paper, the more drugs, antiviral, antibacterial, etc., that have activity, antifungal as well. Then NCI compound library come up with some interesting hits. Anthracycline antibiotic like donomycin, very powerful. It's anti-cancer, very powerful, active against Borrelia persisters, but because of the side effect, its use may be limited. Mitomycin C less so. And then we also did the drug screen against the round body forms. We came up with the drug hits very similar to what we found earlier, except we identified some additional hits like artemisinin, ciprofloxacin, sulfur drug as well. So this is actually the cybergreen PI stain we developed that's better than the backlight dead, live and dead staining, which Monica mentioned. This cybergreen PI assay allowed us to do high throughput screens, and more importantly, allowed us to rank the anti-persistor activity of the current Lyme antibiotics in comparison to the persistor drug candidates we identified. Among the current Lyme antibiotics, it's interesting to note that the cephalosporins are the most active ones. So the lower percentage, the more active the drug is. So interesting, tetracycline as well is more active than doxycycline. And the macrolide's not very active. And our persistor drug candidates are very active, as you can see. So these are some of the other hits. I will not really go into the details because these are already being published: rifamycins, quinolone drugs, antiviral, antifungal, fluconazole, etc. Not go into details. Important to point out that the anti-persistor drugs are different from the current antibiotics active against the growing forms in that they may not have low MIC. They can sometimes have very high MIC. In contrast to the current Lyme antibiotics usually have low MIC, that means very active against the growing form, but have no activity against persisters. And our persistor drug candidates have high activity against persisters, but high MIC, that is poor activity against the growing form of Borrelia. But of course, there are ones that are active against both forms, like donomycin, cephalosporin. So these drugs, the persistor drugs, actually behave differently compared with other drugs in that they seem to preferentially disrupt the cell membrane and inhibit the energy production, as well as the essential oils we found seem to disrupt the cell membrane. And also the damaged DNA, like the donomycin, artemisinin, etc. So we showed that even though we have the persistor drugs, we cannot use them alone. We have to use them in the context of drug combination. And it's, in fact, the triple drug combination, exactly like what I showed you earlier in the context of Yin-Yang. You need to take care of all three different bacterial populations in order to be more effective in eradicating these. So in the Cohen project, our goal is to identify drug combinations that are more effectively eradicate Borrelia persisters, and also develop this oral regimens that's easy to take, because daptomycin, even though very powerful, is an IV, expensive antibiotic. So then we go through these different processes. There are 100,000 of different drug combinations one can test. How do we do it, right? So we have a rule that is according to this Yin-Yang model, that is we know that we need all three types of drugs, and we need to include persistor drug candidates. So with these, then, the idea is to really simplify the current treatment to something like this, so that you get a three or four drug combination to take care of the persistent Lyme disease. So this is just some interim results where we found that you need different drug combinations. We came up with a range of promising candidates, like cefuroxime plus ciprofloxacin plus some other drugs. These, of course, are in vitro studies. Okay, so then we need proper animal models, but I will come back to that. During the process, we also evaluated some essential oils. They came up with very high activity against Borrelia persistens. For example, oregano oil, cinnamon bark, clove bud. They show amazing activities. In the second study, this first one was published a year ago, amazingly has more than 60,000 views. So there's a lot of interest in the use of essential oils. So the second study actually identified additional hits like garlic, allspice, myrrh, haricum, Litsi cubica, etc., as having very good activity against Borrelia persistens. then here it's comparing two different approaches for treating persistent Lyme. One is pulse-dosing, the other is drug combination. In fact, both seem to attack a different type of bacterial population. With pulse-dosing, work only with cephalosporin cytoantibiotic, not working with static antibiotic agents, and then seem to be active against the minor form of the persistence. But for really biofilm-like structures, you can't rely on pulse-dosing. You have to use drug combination without pulse-dosing. So then the animal studies right now is ongoing. Then the only problem with the current mouse testing, the model takes almost 2 years. It's not very efficient. Then we recently developed this arthritis model that works in about 5 weeks. So as you can see here, this show nice swollen in the joints. So we're currently using this arthritis model to evaluate different drug candidates more effectively. So for the future, then, I think we really need to develop a persistent drug regimen that predictably cure Lyme disease rather than get them into remission. Okay, the goal is to cure. Use the C word. I know it's not easy, but I think there is hope. There is promise if we follow up on this type of approach. So for the future, I think we can use this two-tiered approach. One is potentially still the IDSA single antibiotic treatment that seems to work in about 80%. Also, of course, the percentage may vary, but the other is to use the persistent drug regimen, which we're working on. The other possibility is that from the very beginning, one should really use a persistent drug regimen, that is a drug combination approach, so that we don't get a high percentage of uncured patients. So to move forward, then we need to really do clinical trials to properly evaluate the drug combination regimen with the persistent drugs. I need to identify biomarkers for treatment response, need to address the role of co-infection and immune suppression. And above all, we need funding to do the work. And finally, I want to thank people who've done the work, our collaborators, Monica Ambers, Emir Hotzik, and many physicians who have helped provided good advice. And above all, I want to thank Cohen Foundation for providing the generous support for the work. Thank you.